So since we know that, all of these changes are happening on the synapses and in the glial cells in mice, we thought, well, it's very important to try and understand whether this also relates to humans. And this is what I started during my PhD at the UK DRI at the University of Edinburgh with Tara Spires-Jones and Barry McCall. And what we did was start off by using human postmortem tissue. So this is donated tissue that has been paraffin fixed. And we were able, with confocal microscopy and immunohistochemistry, to label all sorts of different markers that we wanted to look at. For example, presynaptic markers like synapsin 1, or glial markers like CD68 and GFAP, CD68 being a lysosomal marker for microglia, and also GFAP being an astrocytic marker. And what we did was to take a lot of images per case from control and Alzheimer's cases. And we were able in 3D to localize synapsin 1 inside these glial cells. So effectively, this is what we term as synaptic ingestion by glial cells. So how much of this presynaptic material is inside the glial cells. And what we found was that both microglia and astrocytes had more synaptic protein inside them. So what we think is happening is that they are eating more synapses in Alzheimer's disease. And we tested this also in two brain areas. We looked at the temporal lobe and the occipital cortex. The temporal lobe being an area affected early on with pathology and synapse loss in Alzheimer's disease. The occipital cortex is affected later on in the disease. But as we're looking at late stage Alzheimer's, both areas are affected. And in both cases, both for microglia and astrocytes, we found that there was an increase in the amount of synaptic ingestion in the occipital cortex, which is very interesting. And it's very interesting because, you would expect that the area affected the most, the temporal lobe, would be the one that has more synaptic ingestion. But it's very possible that because Alzheimer's is a progressive disease, a lot of the synapse loss has already happened before we collect the tissue, because this is, again, at the end stage of the disease. So we might be looking a bit too late, and therefore this is why we see more synaptic ingestion in an area that's more recently affected by Alzheimer's disease. Now, this is the first part of what we did. And the limitation of this is that we don't know whether the synapses are healthy or degenerating, or what mechanisms are leading to these changes. The second part to what we did was that we made these synapse fractions from frozen brains of human cases, either control or Alzheimer's cases. And we tagged these synapses with a red dye that is very good for phagocytosed assays, so things that are being eaten up by cells, as we expect that the synapses are. So this is good because we think that once the microglial cells can eat the synapse, something at the synapse has changed, and we might be able to see what the differences are between control and Alzheimer's cases. So this red dye will fluoresce red once it's inside these glial cells, and we can track this over time. And this is what we did. We used both human and mouse, both astrocytes and microglia in culture. So these were monocultures of one cell at a time, one cell type. And we found that both human and mouse, both microglia and astrocytes, were able to eat more of these synapses from Alzheimer's brains compared to control brains. So it was happening both more and also faster. So this is telling us something about the synapses is being changed and in Alzheimer's disease, and these glial cells can detect this. And the last thing is what I'm doing now in my postdoc, still with Tara and Barry, is what are these signals that are changing? So we had a look at our proteomics that we've published before, and we found that in Alzheimer's synapses, there's a lot of things, proteins that go up and down. However, one of them that we were interested about from what other people have looked at is this thing called MFGE8. And MFGE8 is a protein that others have shown in culture can tag neurons that are healthy ones, or at least living ones, but they are expressing tau. And the microglia eat those neurons up while they're still alive. And so we thought, okay, let's try and block this MFG8 that we see goes up on Alzheimer's synapses. And very interestingly, both human astrocytes and also human microglia in culture ate less of these synapses from Alzheimer's patients where MFG8 was blocked. So that was very interesting. While control synapses, whether they were blocked for MFG8 or not, did not show this difference. There's so many different things that we can look at. So it's really a case of where do we start? So one of the things that we would like to do is that we have an amazing research fellow who started in the lab a while ago now. It feels not that long ago because of COVID. Everything feels like time doesn't make sense. But we have an amazing fellow, a RAD fellow, Research Against Dementia, Dr. Claire Durrant, who is an expert in making these organotypic hippocampal slice cultures where you can study things ex vivo. So we can make these slice cultures from mice, and we can treat them with all sorts of different things. For example, oligomeric amyloid, and test whether this is causing synapse loss in these slices, and see if MFG8 treatment can block the synapse loss ex vivo. So all sorts of things like that are things that we can start testing now.